I'll tell you now about the project that is actually a collaborative project between my lab and Paul Moore's lab. And it has a very, this is the short title, Modular Biotemplates. This is a long title. So the two students in my lab working now full-time on this project are Nathan Carter and Shiken. And this is the big picture of the project, but then I'm going to split it into pieces because it's modular. So what we are really interested in is to combine biological self-assembly, molecular recognition for a purpose of templating of nanoparticles. And what we are really interested in are not individual nanoparticles, but collective properties of the ensemble of nanoparticles. And so the idea and what we think is unique in this proposal is that through this biological molecular recognition and self-assembly, it is happening really on the nanometer scale. We have a possibility to pattern this nanoparticle array with a single nanoparticle resolution. And so this is going to give you much better resolution than the top-down approaches that are currently used. And so the strategy is depicted here in this little picture, where in my lab we design these are de novo protein sequences that have self-assembly properties. Within these protein sequences, we can engineer specific peptide binding sites. On the other hand, we can take the nanoparticles, we will modify them with the peptides that specifically bind to these proteins. And now when we mix nanoparticles and proteins and assemble thin films out of this material, depending on orientation of the peptide binding site within the self-assembly protein, we can make a thin film in which nanoparticles will array in different geometries. And our hypothesis is that depending on the geometric arrangement and orientation of nanoparticles in respect to each other, these thin films will now have different collective properties that we can see in their absorption properties and light properties. So this is the protein design part of it, and this is what we consider, why we consider the system to be modular. So we design modules that are binding peptides, and then separately design so-called spacer modules. And through appropriate combination of the B modules and S modules on a gene level through recursive ligation, we now can make the superhelical proteins that will have a peptide binding sites with different kind of orientations. And the first generation of the proteins that we made that we're looking here are these superhelical proteins. They're about 18 nanometers long, and they have a peptide binding site 120 degrees offset from each other along the superhelical axis. So we went and we looked into self-assembling properties of these materials, and we can make thin films out of them. They are pretty large. You're looking about 5 centimeters big film. They are uniformly birefringent under the cross polarizers, which tells us that all the superhelices are oriented in the same direction. So we really look at the long term or the long range orientation within this film. So we looked at the thermal properties. We can heat these films up to 160 degrees Celsius without interrupting the alignment between the film. I also want to mention that these proteins are very stable. They don't lose a secondary structure until way above 100 degrees Celsius. We looked at the mechanical properties of the film, and we compared Young's modulus of our films with different biological and non-biological tissues, and we are somewhere on the order of magnitude of what the collagen would be. I also want to emphasize that this is all non-covalent self-assembly. There's no entangling. This is all molecular packing within the film. This is really also nice property. So these films are transparent, and we can measure translucence, which is almost 100%. Okay. So our first goal in this project was actually to make these proteins and characterize self-assembly, and so we kind of showed that we achieved the first goal. And so then this project kind of took a life of its own. And I got a student in my lab who has experience in solid-state physics and in nanoparticles, and he wanted to take a look if these proteins by themselves can pattern different morphologies of nanoparticles because they have these interesting anisotropic shapes. And I also want to mention that there are no cysteines in these proteins. People usually associate sulfur with patterned wall, so there are no cysteines in these proteins. And so what we do is we mix gold precursor in one of the good buffers. So we're looking at the heat here. This is totally in the aqueous solution at a pH 7.4. And by varying concentration of the protein and ratio of the protein corresponding to the gold precursor, we start making gold nanoclusters that have different morphologies. And these nanoclusters have different optical properties as well. And so this is where we are with these two modular pieces. Okay. So this year, what we are planning to do is now where it's actually taking quantum dots, mostly looking at the cadmium sulfide and cadmium selenide. We are developing this high throughput phase display technology where we will now pull out selectively peptides that bind both our protein and semiconductor material. We will then do ligand exchange to create these nanoparticles that will specifically bind to the peptide. Then we plan to do layer by layer self-assembly, where we will create this type of film with the current geometry of binding sites that we have. We hypothesize that we will make this type of films where this would be the orientation of the semi-production nanoparticle, the quantum dot. This will be in a plane of the film, and this will be the cross section in a direction of the film graph. So take-home message is that we're trying to develop a bottom-up platform to make this kind of nanoparticle arrays with hypothesis that the geometry and interaction now between these nanoparticles and their collective properties will give different optical properties to the developed material. I actually have a pending proposal with the Department of Energy for biomolecular materials in this, and we plan to apply to both UV and NSEP for this in the future. And I'll take any questions. Thank you.
We have time for a question. Thanks, Tim. All right, moving on. I think you're up next.
Perfect time.
Yeah.
Well, thanks everyone. I'm going to go over really a mixture of the two seed projects that we have where we're looking at the development of artificial cochlear systems. And just to acknowledge my collaborators, Wally Grant, Pablo Terrazaga, and Ralph Mueller are collaborators on the program. And this gives you an overview of some of the background and significance in terms of the medical need. But the real question that we're trying to focus on is, can we develop artificial hair cells that mimic the sensitivity and frequency selectivity of natural hair cells that are in the cochlea? And our research objectives for this seed program were to fabricate and test an artificial hair cell. We had some prior work where we had developed these artificial hair cells on the mesoscale. And so part of our research objectives for this work were to reduce the length scale associated with how big we could make these hair cells, measure the mechanoelectric response and try to understand the physics to a greater deal of accuracy, and then develop a test setup where we took an artificial hair cell system and measured the electrical response to various mechanical inputs. So this is our progress. I want to really highlight the work that Wally Grant's done. Him and his student Miles Dunlap have been great collaborators on this program. But let me show you what this is. This is a picture not of a hair cell. What we're developing here is two droplets. And each one of these droplets has a salt solution in it. And this is a micropipette that pierces the droplet. This is another, it's hard to see, but this is another micropipette. And this droplet is surrounded by a lipid monolayer. And so is this droplet. And then at this interface, it forms an artificial bilayer that's about 5 nanometers thick. And the function of this bilayer is essentially to act as a transducer. And this is what we had demonstrated prior to this work. But this work has really given us a lot better insight into the physics associated with the transduction properties of the bilayer. And what we're just showing here is that we can create these bilayers. One of the goals was to create these bilayers at smaller length scales, and Miles has achieved that. This length scale here, I apologize, I don't have a size, but this bilayer is probably about 30 microns. 30 microns in diameter, and we have made ones that are on the order of about 10 microns in diameter. So we have been able to reduce the size scale of the bilayer. The figure that you're seeing here is a demonstration of the fact that the bilayer area change correlates to the current output generated by the transducer. We had hypothesized this with some of our previous work, but the work that Miles has done has demonstrated that there's a direct correlation. And this blue region that you see here is actually an electrical measurement of the bilayer area. And the green is, the blue is the current generated by the bilayer as the area changes. The green is a measurement of the bilayer length from a visual measurement. And so what you can see is that the change in the length of the bilayer, which obviously we could correlate to area change, directly correlates to the current that we're getting out of the bilayer as it's changed dynamically. And so this, I think, verifies our original